Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
In a first for the genetic toolset known as CRISPR, a recently discovered protein has been found to act as a kind of multipurpose self-destruct system for bacteria, capable of degrading single-stranded RNA, single-stranded DNA and double-stranded DNA. With its abilities to target so many types of genetic material, the discovery holds potential for the development of new inexpensive and highly sensitive at-home diagnostic tests for a wide range of infectious diseases, including COVID-19, influenza, Ebola and Zika, according to the authors of a new. Using a high-resolution imaging technique called cryo-EM, the team discovered that when this protein, named SARS-12A2, binds to a specific sequence of genetic material from a potentially dangerous virus, called a target RNA, a side portion of SARS-12A2 swings out to reveal an active site, similar to AS. Then, the active site starts to indiscriminately cut any genetic material it comes into contact with. The researchers discovered that, with a single mutation to the SARS-12A2 protein, the active site degrades only single-stranded DNA, a feature especially useful in developing new diagnostics tailored for any of a wide range of viruses. A test based on this technology could theoretically combine the best features of PCR-based tests that detect genetic material from a virus, high sensitivity, high accuracy and the ability to detect an active infection, with the best features of rapid at-home diagnostic tests, inexpensive to produce. It also would be easily adaptable to any new RNA virus. If some new virus comes out tomorrow, all you have to do is figure out its genome and then change the guide RNA in your test, and you'd have a test against it, said David Taylor, an associate professor of molecular biosciences at the University of Texas at Austin and co-corresponding author of Th. Such a diagnostic would still require separate work and probably involve collecting saliva or a nasal sample from a patient to be mixed with the team's modified SARS-12A2 protein, the piece of guide RNA that acts like a mugshot to identify a specific virus, and a fluorescent probe designed to light up. CRISPR is the name for a set of tools that occur naturally in bacteria, but which scientists have adapted for use in gene editing. This is the first CRISPR protein that has been found to degrade such a wide range of genetic material. SARS-12A2 basically grabs the two ends of the DNA double helix and bends it really tightly, said Jack Bravo, a postdoctoral fellow at UT Austin and co-first author on the paper. And so, the helix in the middle pops open, and then this allows this active site to destroy the bits of DNA that become single-stranded. This is what makes SARS-12A2 different from all the other DNA targeting systems. The paper's co-corresponding author is Ryan Jackson and co-first author Thompson Hallmark, both of Utah State University. The other co-authors are Bronson Nagel of Utah State and Chase Beisel of the Helmholtz Center for Infection Research and the University of Würzburg in Germany. Structural data were collected using the cryo-EM facilities at the Sauer Structural Biology Laboratory at the University of Texas at Austin. Taylor, Bravo, Hallmark and Jackson are inventors on a patent application covering modifications to the SARS-12A2 protein that enable it to cut only single-stranded DNA and for its use in diagnostics. The UT Austin Office of Technology Commercialization is managing the intellectual property and working to find industry partners that can help realize the potential of the technology. This work was supported in part by the National Institute of General Medical Sciences of the National Institutes of Health, the German Federal Agency for Disruptive Innovation, the Welch Foundation, and the Robert J. Kleberg Jr. and Helen C. Kleberg Foundation. David Taylor is a CRISPR scholar supported by the Cancer Prevention and Research Institute of Texas. A companion paper in the same issue of Nature describes the biological functions of SARS-12A2, while the paper described in this news release describes the mechanisms by which the protein accomplishes them. Story source. Materials provided by University of Texas at Austin. Note: content may be edited for style and length. Journal references: Jack P. K. Bravo, Thomson Hallmark, Bronson Nagel, Chase L. Beisel, Ryan N. Jackson, David W. Taylor. RNA targeting unleashes indiscriminate nuclease activity of CRISPR SARS-12A2. Nature, 2023, doi, 10.1038, S41586-022-05560W. Oleg Dmitrenko, Gina C. Newman, Thomson Hallmark, Dylan J. Kaiser, Valerie M. Crowley, Elena Violetto, Ioannis Mougiakos, Katerina G. Wandera, Hannah Domgaard, Johannes Weber, Thomas Godin, Josie Metcalf, Benjamin N. Gray, Matthew B. Biegman, Ryan N. SARS-12A2 elicits abortive infection through RNA-triggered destruction of dsDNA. Nature, 2023, doi, 10.1038, S41586-022-05559, 3. Cite this page. University of Texas at Austin. How a CRISPR protein might yield new tests for many viruses. Science Daily, 4 January 2023. ift.tt/enfraha5. University of Texas at Austin. 2023, January 4. How a CRISPR protein might yield new tests for many viruses. Science Daily. Retrieved January 4, 2023 from ift.tt/enfraha5. University of Texas at Austin. How a CRISPR protein might yield new tests for many viruses. Science Daily. ift.tt/enfraha5, accessed January 4, 2023. Thanks for watching. Please subscribe my channel for more videos.